So just want to put my name in there. I'm kind of, I think I'm the other side of the country here. I'm in Virginia. So I think where most of you guys are in others are in Oregon. My dad's from Oregon, actually was.
Nice.
Yeah, and I'm also invading. I'm actually a chemistry person, but I've been using the fluorescence for the drink, the energy drink glowing. The students loved it. And I've been using some of the, we take out in organic, we take out pigments from the plants, and then we look at them in, with the spectroscopy. So it's all, really good. So I'm just curious to see what new stuff you guys are coming out. We use all of you guys' stuff, I mean, we are like your best customer, and we just like ordered like a ton of stuff. I can't believe how much money they gave us. So I wish they'd, give us some of it, but they're really willing to spend it on things. But yeah, so so I'm really excited. We always are watching to see what new stuff you guys are coming up with. So so it's really excited to see some of you all.
That's awesome. I'm so happy to hear you enjoyed the energy drink lab and that you used that for that.
They loved it. I mean, they were like running around. I was like grabbing people out of the hall and showing them how they could take this, and it was like glowing, and
awesome.
And, yeah, and they were all, they were all like, wow, wow, wow. So then we were running around trying to find all the energy drinks that actually have riboflavin in them, because it's a lot of them. Red Bull doesn't have it in anymore.
No, yeah.
So we had to be really careful about that. But yeah, that was a great idea.
Oh, good. Yeah, I wrote that lab, so I'm always happy to hear how they turned out in the actual classroom. So thank you for sharing that.
Right, you should. Yeah, I'm so happy. It's a really good idea. So
awesome. All right, John, we'll get started here. I see that quite a few of you have joined us. So welcome to our webinar, Color and Light Investigating Plant Pigments. I'll just go over a few things real quick and then pass it over to John, who's going to be presenting today, and Melissa, who is going to be helping answer your questions. So first off, I'm going to launch a quick poll just to get some basic information from you. So while I'm going over these items, if you don't mind answering those two questions, that would be great. So we are recording the webinar, and you will get the recording and also links to a shared folder with resources in it for you that I'll share in the chat in a few minutes. And you'll also get a certificate of attendance for joining us today. And so we will be answering questions throughout the session. So feel free to put those in chat. We'll be monitoring those. And if we happen to not get to your question, we will follow up with you afterwards. So with that, I'm going to pass it on to John.
Great. So let's see. All right. So
and it looks like, John, that sorry, the poll, I'll show the results here for you. Are you able to see them?
Yes. Okay. Yep, great. Perfect. Great. So let's see here. So I'm just going to go over the agenda really quick, introduce you to me, and then talk about the lab, and then we'll start playing with the fun stuff. So really what we're going to be doing is I'm going to be talking about spectral analysis, which is a software title that you can use with your spectrometer. It runs on any platform, but everything that I'm doing could also be done with a LabQuest, could also be done with Logger Pro. None of this is specific to spectral analysis. You just need to have a spectrometer and then use any one of those titles. All the labs are written that way and they will all work. There's an introduction to plant pigments. This is experiment 14 from our inquiry biology lab, or our book called Investigating Biology Through Inquiry. And then I'm going to do a hands-on demonstration. If you're not that interested in inquiry, don't worry, there are non-inquiry versions of these two labs. So basically, the idea for this inquiry lab to look at plant pigments and olive oils and all sorts of other crazy things that you can do is developed from our advanced biology book, which looked at the classic chlorophyll extraction, and then also looked at olive oils, which are really interesting because olive oils contain chlorophyll. So there are non-inquiry versions of this activity that you can find on our website. And so once you get the the slide deck, you can go back here and find this if you ever need it. But I've also put all of these labs in the folder that you have access to. And then hopefully we'll have some questions and answers, and then you'll see how to view the webinar. My name is John Melville. I got my biology degree, my BA in biology and psychology from Sonoma State. I got a PhD in zoology from Oregon State University. And then I did a postdoc at University of Oregon. My background is in zoology and neuroscience. And then I taught majors and non-majors biology at a little small college called Wartburg College. But I also taught at a community college level as well. And I've taught pretty much everything for biology. What a lot of people don't know about me, and one of the reasons why I love this lab so much, is I actually started off as a marine biologist, which most biology majors do. Everybody wants to study marine biology and then they do something else. But I took like a lot of marine biology courses, advanced courses in phycology, the study of algae, invertebrate biology, and things like that. And so there's an activity at the end that I can show you about how to, if you want to, look at evolution using algae powders, because algae have different types of chlorophyll and other photopigments, accessory pigments, than plants do. Just remember, there's a Q&A feature. Drop your questions in chat. Feel free to ask them. Someone will be there to interject and tell me, hey, John, here's a question for you. And then we'll have other questions at the end. Great, saw that there. The other thing that I just wanted to remind everyone is the big thing that we're here to talk about, right, is this graph that you see down here, the chlorophyll absorption spectrum. Normally, when we do this lab in a biology class using a Spec 20, it takes you about an hour to 2 hours just to show this graph. And what I wanted to let you know is, right, what we're really interested in is chlorophyll A and chlorophyll B, and they have specific peaks at 425 and 662, and then chlorophyll B, the other photosynthetic pigment in plants, is here. And this is just a Wikimedia Commons page where if you want to see the actual spectra of chlorophyll A and chlorophyll B separated out, not together, you can find that as well. Now, the reason why I love doing this lab is because, like I said, this used to take me about an hour to do with my students, and then they would hand create a graph. They would literally be drawing it by hand. Using spectral analysis, you can do that in under a minute. And so what usually happens when you're doing this lab is most college professors say, well, what do I do now? And so what we came up with is you can do all sorts of things. You can look at different plant pigments. You can look at olive oil. You don't have to just spend all of your time teaching students how to make a graph. You can teach them about photosynthesis, pigments, the pigments that are involved in the process, and even look at fluorescence. But don't forget, you don't have to use spectral analysis to do this. You could use a LabQuest 3, LabQuest 2, or Logger Pro to do these labs. But the key things here is that it's fast. You can collect full spectra really fast. And what I'm going to do is compare spectra from extracts of spinach and berries. We're going to compare different types of olive oil, and then we'll investigate fluorescence and maybe talk about evolution a little bit. So just once again, this activity is from experiment 14 from Investigating Biology Through Inquiry. That's in our inquiry biology book. I'm going to be using this spectrometer right here, the GoDirect SpectroVis Plus, and we're going to be using spectral analysis. And there are also non-inquiry versions that we have adapted. They're not in this book, but they're in the folder that you have. And if you also click on this link right here, there's a website where the non-inquiry versions are as well. All right, so what we're going to be doing is we're going to be recreating this graph down here that you can see, where I've got some spinach and some cherry, and we're going to be looking at an entire spectrum from 400 nanometers to 900 nanometers. And the key points to just remember, if you haven't used one of our spectrometers before, is the solvent is always going to be the blank. So if you're dissolving something in water, water is a blank. If you're dissolving it in isopropanol, isopropanol is the blank. And then to get really good resolution of these peaks, you want to make sure that your peak absorbance, if you look over here, absorbance, you want to make sure that's below 1. I don't know how many times someone has called us saying that they have a problem with the spectrometer, and I asked them to send me a graph, and they've got an absorbance of 3 or 4, and I'm like, you need to dilute, just dilute with your solvent, and you'll get great spectra. So those are the 2 key points that I want to bring up. All right, so just to get started, let's just take a look at spectral analysis, and let's just take a look at some food coloring. So let's just take I'm gonna click on spectral analysis here. And then I just want to show you the spectrometer right here. So I've got a spectrometer right here. Hopefully you can see that. It's plugged in over USB. You can also use it over Bluetooth. I just like using it over USB. And then what I have over here is I have some cuvettes that I've already made. Let's just take a look at some things in water first. So I'm going to take water as my blank, put it in the spectrometer. Just remember that the light source is here. So light goes this way. So if you look at these cuvettes, there's a clear side to the cuvette. There's a ridge side. You want to make sure the clear side is facing the lamp and the slit, which is on this side. There we go. And I'll switch over here. Now I'm just going to launch spectral analysis because I want it to be like we're starting all over. And you're given all of these choices when you start: absorbance, transmittance, fluorescence, emissions, and then there's a really cool advanced mode. Let's just go to absorbance, and I just want to collect spectra. That's all we're going to do. I just want to look at spectra, pretty spectra. So we're going to look at wavelength versus absorbance. And then I'm going to calibrate the spectrometer. Normally, if I were to hit collect, it should tell me that I need to calibrate. Let's see here, stop. Just going to let it warm up here. There we go. I'm going to hit skip, finish calibration. Oh, calibration failed. Hold on. I'm just going to reboot. Relaunch spectral analysis. There we go. And the reason why we're waiting for it to warm up a little bit is it's got an incandescent lamp and then a series of LEDs, and we want to make sure that all of those come up, heat up to a certain level before we actually do the calibration, or we can see odd spectra. But as you can see, you don't have to wait the entire time. You just have to wait 20 seconds. I'm going to skip, finish calibration. There we go. So let's just take a look at red food coloring, just as a start. So I just placed the sample in the spectrometer. I hit collect, and then I'm still collecting spectra. I'm going to hit stop. And I just want to remind everybody, right, that what's really great to talk to your students about when you're teaching about pigments, photosynthesis, et cetera, is we're talking about absorbance. And absorbance is what the pigments absorb, not what they transmit. So if you see in this case, it's absorbing mostly blue and green, and it's not absorbing any red. And that's why it looks red. It's because it's transmitting red, it's absorbing in the other ones, right? So this is red. So that's red food coloring. Red food coloring is going to absorb in the blue and the green. Let's just take a look at green food coloring. I want to make it a little bit more interactive. So where is it going to absorb if I use green food coloring? Somebody tell me. Green food coloring.
In the red?
It'll absorb, it's going to absorb in the, you can see it's going to absorb in the red, not quite much in the green, but a little bit there, and then mostly in the blue, right? And then what about like blue food coloring? it's going to absorb mostly all in the red and none down in the blue. And I think, I know we're not able to, I can go over here in spectral analysis, I think, data set 1. I can rename them. And I can say red. Green. Blue. And that's nice because if I go down here into graph options in the bottom left corner and I say graph legend, you can see it's gonna, sadly, they're not the right colors there, but I think I can even change those. So I can change this one to red. I can change green to green. And I can change blue to blue. Oh, there we go. Red, green, blue. And you can see they're not absorbing where you would think that they would absorb. So it's a great way just to teach students about what absorbance is, right? So questions so far? All I'm doing in this case is I am collecting spectra. I can label those spectra, and then I can also inspect those spectra. So you see if I drag along here, I can find the peak amplitude and absorbance, and down here in the bottom of the graph on the X graph, I can also find out what the wavelength is, which is important, right? So peak for red is at 501. And then it looks like green food coloring is really blue food coloring with probably another food coloring if you look at these spectra. So questions, everybody's fine with that, pretty bored? Yep, get it, no problem. All right, so then let's just do file new and let's just take a look at some plant pigments. Now, the cool thing about plant pigments that I want to show you why I love doing this lab is here are some plant pigments. This is spinach, just extracted with isopropanol, 70% isopropanol. This is raspberry, extracted with isopropanol. You don't have to follow the directions in the book, use a mortar and pestle, use petroleum ether or anything like that. You really can just use 70% IPA and it works great. And I take this lab on the road. My friend Mike Collins taught me how to do this. We took this on the road everywhere, doing workshops all over the country because it's so simple to do. All you need is coffee filters and a coffee filter holder, something to hold the coffee filter. And then the other thing that is important is you need some isopropanol. And then you need, oh, where are they? Where are my bags? Here we go. You just need a Glad bag. So what you do is you just take a nice handful of spinach, toss it in the sandwich bag, pour in a liberal amount of isopropanol, and you just squeeze it with your hand, pour it through the coffee filter, let it sit and collect it up. And if you have 3 coffee filters and a bunch of solo cups, you can make plenty of material for your students in like 30 minutes. Or what's even more fun is you make your students do the work. So you have them take a look at it, you have them pull out the berries, you have them mush them up, etc. And then the nice thing is you have fruit to take home too, if you want to. So it works great that way. So let's just take a look at some spectra here. So let's take a look at, I'm going to take a look at, so remember, I'm going to recalibrate because these are all with isopropanol. The solvent is isopropanol, so I'm going to calibrate that way. So my blank is going to be isopropanol, and then I'm going to show you a raspberry and then spinach. Great. So let's take a look at raspberry. Let's take a look at the color raspberry. Raspberry is nice and red, right? kind of like red food coloring. So let's take a look at it here, go back. Ah, beautiful. So you see it's absorbing mostly in the blue and the green and none in the red. That's just a classic raspberry extract. All right, what about chlorophyll? What is chlorophyll gonna, what is spinach? Sorry, what is spinach gonna look like? There's this nice kind of olivey green.
Any ideas?
It should look like
Blue and red.
it should look like the green.
It should look like the green, or hopefully, hold on. Oh, where'd I go? Go back there. Hold on. Start slideshow.
Or it could be the blue and yellow, I guess.
I'm actually hoping it looks just like this graph, right? Because that's what we want to make, is this graph right here. So, but you could see in that graph, it's mostly blue, a little bit in the red, not a lot in the green. Leaves are green. That's because they're transmitting green and they don't really absorb that wavelength. So let's take a look. There we go. So if we take a look at this here, you could see that this right here, let's just change these colors here, right? Let's make data set 1 red. Let's make data set 2 green. And then I can even label them over here, rename. Raspberry. Here, rename data set spinach. And then I can turn on graph legend, and you can see there's raspberry and spinach. And if you look at this, you can see here's a peak at 634. That's probably mostly, let's just take it back and look here. See, so peaks at 425 and 622, 662 are chlorophyll A.
Does green food coloring produce the same wavelengths as chlorophyll?
No, that's what's great, right? Is if you look at green food coloring, I'll just put it in here. Remember again that green food coloring, this is going to be off because the solvent is water, but let's take a look at green food coloring, right? So there's green food coloring. It absorbs some in the blue and a lot more between 600 and into the red, but it's very different than chlorophyll. Chlorophyll absorbs a lot in the UV, and then it also has this really cool little peak here that's very specific. This 660 peak, and then these two peaks. And this is a mixture of mostly chlorophyll A and chlorophyll B and probably a little bit of carotenoids. So questions? Super simple lab, easy to do, students love it. You can look at all sorts of pigments. You can look at rose petals. You can look at other pigments that are water soluble, like from beets. Those are water soluble. That's anthocyanin. There's a pH shift to it. There's all sorts of things that you can do if you want to begin to look at inquiry. How many of you have looked at or know that chlorophyll fluoresces or looked at chlorophyll fluorescence before? Yeah, I know. So that's another really cool thing that you can look at too, that maybe we should do, is chlorophyll has fluorescence, which I think is one of my favorite things to look at and do. If I were to, don't know if it's going to work, but it's probably not dark enough, but let's just see if I can get it to work. So there's that. I've got a little blue view here. And you're probably not going to be, if you were looking at this, it would be very cool. It's glowing a nice bright red underneath there. There's probably too much reflection and stuff for you to see it there, but it's actually pretty neat if you saw it in the dark. I just can't get up and change everything. But that is a another cool thing that you can do is you can look at fluorescence. So for example, let's just take a look at, or we can look at olive oils. We can come back. Why don't we do, don't save. Let's just take a look at fluorescence because I've already got the samples out here right now. All right. So let's just take a look at green food coloring. I'm going to put some green food coloring in here. I'm going to change the wavelength of excitation. Let's look at the 405 LED. And so in fluorescence, what we're doing, there I'm back. What we're looking at fluorescence is we're taking a wavelength of light and we're trying to excite the sample, and then it should glow or emit in another wavelength. And I'm a former fluorescent microscopist. I used to play with fluorescent molecules all the time. They're great for labeling. They've been super important. So I love fluorescence. And this is also one of the reasons why chlorophyll is so wonderful at doing what it does, taking energy from the sun. It's because it can absorb it and transmit that energy. So I'm just going to take a look at this green food coloring and let's see if we have any fluorescence at 405. That looks like a flat line. The 400 that you see right here, that's actually from the LED itself. So that green food coloring is not doing anything. Let's take a look at some spinach extract. And the same thing, what's really cool here is if I were to look at this, if the students were to look at this or you were to use a UV lamp or anything, you would see that it's glowing bright red. And you can see that right here. There's this really bright red glow right here, or peak, very sharp peak. And that is the fluorescence from chlorophyll. Now this can get really important if you want to analyze olive oils, which we can do next, because looking at the fluorescence of chlorophyll is a really good indicator of the quality of the olive oil that's there. The University of California, Davis did an entire study on olive oils, and lo and behold, what they found is that for them, olive oils from California were the best, not because California olive oil was the best, but because those were the closest to them. So the freshest samples are going to be the freshest. And there's a whole industry of adulterating olive oils and mixing olive oils and things like that. And that's why one of the super cool things that you can do is if you invest a little bit in some olive oils, these are all different types of olive oil here. And there are different types. I have like, this is pure, it says pure olive oil. And then there's extra virgin, and then there's extra light. And these are all the same brand. So you could compare the same brand of extra light, extra virgin, and pure. The difference between those brands is how they process them. So extra virgin olive oil is cold pressed, and light olive oil is processed by heat. And when you heat up olive oil, you destroy the chlorophyll. So that's why it's lighter in color. I also have another set of olive oils here. If you look over here, these are from Kroger, pure, extra virgin, and light. And then another really neat thing that you can do is I have an olive oil from Italy, an olive oil from California, and then another olive oil that it doesn't say where it's from, but if you read on the back, it'll tell you that it's from Italy, Spain, Tunisia, and Argentina. So there's lots of ways that you can make this activity inquiry just by looking at olive oils too and looking at the spectra. The nice thing about looking at olive oil is you don't really even need a solvent. You can use vegetable oil as your solvent, or you could use water or isopropanol, it doesn't matter. So questions so far before I move on?
This isn't really a question, but I just want to say there is a video out there on YouTube. One of the students somewhere in biology, he actually writes on a leaf. I don't know if he writes on it or if he puts something over top of it so it doesn't absorb, I forget. But then he takes the leaf in a dark room and you can actually see the letter. It's actually, I think it would make a really, I will replicate it at some point because it'd be a great demo. But yeah, it's pretty impressive.
Yeah, the classic demo that I used to see when I was taking my biology class. So someone just did an isopropanol extraction of spinach, and then just brought a UV light. You just bring a UV light, and you turn the lights off, and you just bring the UV light close to your sample, and it glows bright red, and everyone goes ooh ah.
Right.
So it's super simple to do with a UV light or even just a blue lamp.
So I do have one other question. So what is the range on the wavelengths on the spectra?
That is a great question. So if you take a look at the spectrometer, it really only has two wavelengths. It's got a 405 and then a 500. So if you look over here.
For the fluorescence, and then, but don't you guys have a new one coming out that does more than that?
Oh, yes. We have.
Because I think we've got some on, does more for the fluorescence, and then, and just the regular wavelength. I think, don't you have one that goes down to somewhere close to 200 or, yeah.
The super fancy, hold on. We have a UV Vis, which works great, which goes down to, I believe the lowest is 220. Is that right, Melissa? 220. And then we also have, if you want the, I like to call it the Ferrari of spectrometers. You call it the Lamborghini, you call it whatever you want. But the fluorescence, the Vernier Fluorescence Uv Vis. So it's a UV Vis spectrometer. And then you can see these cartridges. Right. These cartridges are all different LEDs. And I believe that it comes with the 375, 450, and 525, but there are other LED cartridges that you can buy.
Okay, cool.
Yeah, so it's.
I was not aware of that. That is a new thing, I think. So
it's been out there for a while. This one's, I think, finally the Go Direct. That's new this year, the Go Direct one. But we had an older one. But it is what I like to call I don't want to call it the Cadillac. I prefer Ferrari, so I would just call it the Ferrari of spectrometers. So it's many different interchangeable LEDs, a UV light source, so it's visible and UV. So it does all of it. It's a very nice device.
Yeah. So you guys are moving pretty much away from LabQuest. Is that the case?
No, this device works on LabQuest. Okay.
And like the experiments are still, some of them are still written with directions that go to LabQuest. We're just in the process right now of changing from, we just ordered a bunch of equipment from you guys, and we're changing from using the LabQuest to getting, we got funding to get computers and then go to Logger Pro. But we still are, all the experiments are still on LabQuest. So
what's great is if you have a book, so I don't know if you're aware of this, let me just show everybody really quick.
We have all the books.
Okay, so then you have access, you should have access then to all of the instructions. So one of the things that we tell a lot of people that they may not know about is if you go to the top right-hand corner of our website, and if you create or log in, what you have access to are all of the downloads that are associated with you, and then also all of the lab book electronic resources. So, and these instructions, as they're getting updated, will have all the instructions that you need. So like if you have investigating biology through inquiry, where is that here? If you download the electronic resources, it has instructions for LabQuest, Logger Pro, spectral analysis if you have it, and then the Go Direct sensors as well. The only place where it gets a little bit tricky is our Go Direct sensors can work on a LabQuest. And so there aren't specific instructions for Go Direct sensors on a LabQuest, but in most cases, a temperature probe is a temperature probe. Once you connect it to the LabQuest, the instructions are going to be the same, very similar. And I'm sure Melissa can correct me if I'm wrong there, but in most cases, it should be straightforward. The only case where it's not really straightforward would be human physiology, and that's just because the sensors are very different how they work.
All right, thanks.
Great. So what would you like to see next? Should we take a look at some olive oil? Interest? Yes, no? Boring?
Yeah.
All right.
Yes.
All right, so what I'm going to do in this case is let's start over. And I'm going to do absorbance versus wavelength. And I'm just going to pull over here some extra light, extra virgin, and original pure olive oil. And while it's warming up, I'm going to make those 3 samples. And while it's doing that, I will also show you a trick. Oh, don't want that. Hold on. There we go. I'm just going to show you a little trick. Oil is pretty viscous. So one of my tricks for looking at olive oil or putting olive oil in a cuvette is just clipping off the tip of your pipette, and it's going to be easier to transfer the olive oil. So here is some extra light. You also could just pour it in there. The really nice thing also about this spectrometer, I want to give out some major kudos to Melissa, because Melissa is the project manager, designer, etc. for our GoDirect SpectroVis Plus. And when it went from the SVis to the GoDirect, it's super nice. The volume that you need to put in here is so much less than you normally would need. You need like only about 1/3 of the cuvette. Originally, you needed 3/4. So you can save a lot of reagents once you know that. So here is some extra light. Here is some quote unquote pure.
What is the best way to clean out your cuvettes after they've had the oil in them?
Oh, that's tough. I tend to just throw them away after they've had oil in them, just because they're going to be so hard to clean. Whereas anything else that's water soluble or isopropanol, etc., I just wash and then throw in a little bit of bleach. You could try washing them in like some Dawn liquid detergent, letting them sit there in warm water, you know, and just rinsing them a lot to see if it comes out. But oil is weird that way. All right, so I'm just going to use, pardon me. Go back to spectral analysis. You can already see that these are different colors, right? So let's go back here to spectral analysis. I'm just going to calibrate the isopropanol. I could use vegetable oil, but let's just use isopropanol. So let's take a look at extra virgin first. There we go. So there's extra virgin olive oil, and what you hopefully that you should notice is that this is chlorophyll A and this is a chlorophyll A and chlorophyll B extraction right here. Like if you want to just show chlorophyll A and chlorophyll B and not even have to use spinach, extra virgin olive oil works great. There's your 660 peak. This 610 peak is actually a little bit from chlorophyll A2. A chlorophyll B peak would be over here around 640 to 650, but it's this peak here, the 480 and then the 450 that are critical for chlorophyll. They're also showing chlorophyll A and chlorophyll B. All right, so let's just label that really quick. So let's just say this is EVO. And I'm also going to say this is Star, because that's the brand. And then let's take a look at quote unquote pure. And you can see it's got not much of anything there. And by the way, these all both cost the same. I mean, and then let's take a look at some light olive oil. Extra light. You can see it doesn't even have this little bump here, right? So the There's a teeny bit of that stuff there, but you know, like you can easily see the difference between these three different types of olive oil, no problem. And extra virgin has the most, pure is pretty bad, and then extra light is pretty bad. Any questions there? Because we can also take a look at fluorescence pretty quickly too. I could turn around here and let's just take a look at fluorescence for a new experiment. Let's take a look at fluorescence as a function of wavelength. And, oh, that's really, okay, so let's just switch to the 450, 405. And let's take a look at extra virgin first. I can already tell that it's glowing a nice bright red. I'm going to hit collect. Now, one of the things that's nice is if you take a look at this peak, you see how it's sitting at like 0.2. I can adjust the dynamic range to make it so that I think this is probably going to one that's going to be the highest. So what I can do over here is if I tap on the settings icon, the gear, I can increase the integration time. The integration time is a lot like an old school camera for how long you're collecting light. So let's just increase to 100 milliseconds and see what happens. There you go. I'm at like 0.5 average units there. That's probably pretty good. Why don't we just say 200? Oh, now I'm above 1 and I flattened out that peak. So I'm going to come back and say, let's just do 150. What I'm doing in this case is I'm bracketing the sensitivity of the fluorescence response to see where these peaks are. So I think pretty much that I know extra virgin is going to be the tallest. So I want to make sure it's not peaked out. So I've got a nice sharp peak, goes to 0.8, and then I'm going to leave the integration time alone. So that's extra virgin. Let's take a look at pure. Oh, so you can see there's some, but there's also this other stuff that's in there. That's interesting. There's some other fluorescent peak that's over here. That's kind of odd. All right. And then let's just take a look at light.
So you said that one that you just did was pressed with heat, right? Or whatever heat's used to get.
Yeah, it's pressed with heat, and also it's just called pure, is what it is. That's the difference.
Would the heat take something else that's in the olive out?
No, I think what it is it's just degrading, and then you've got I don't know what this other stuff is that's in there. I've never kind of seen a peak. It's actually kind of small, but it could be just a ripple in the background. But it's interesting that it's not there for the yellow one, and that it's there for these other two. It could just be part of the degradation process. What I have seen in some cases, and I don't think I see it here, is if I switch this LED to 500. It's kind of glowing a bluish color, but that's from the 400 LED. In some cases, I have seen olive oils that are adulterated with things that look very similar to riboflavin. So like they'll have a greenish color, but then they glow bright yellow. But I just think that's part of the great degradation process of it being heat pressed that you see. But you can see that even with the light, there's very little chlorophyll left. Right, so with the red peak here, there's no peak there at all. So questions, that was just looking at extra virgin, star extra virgin, star original pure, and then extra light.
Did the blue line when you did the regular absorbance, did that have chlorophyll in it? Was it, there's a bump there?
Yeah, well, it's super easy to do this over again. So let's
I was just
say,
curious.
don't
It was like, it
say-
must if it's fluorescing. But.
Yes. Well, let's just do it over again really quick. Well, that's what's so great about this is you can, since it's just comparisons, you can quickly look at lots of different samples.
John, why don't you show them in advanced mode so they can see the absorbance and fluorescence side by side?
Oh, that's good. I didn't want to go all Stoke shifty on them, but we can do that. Okay, so.
I just think it's easier, frankly, than having to go back and forth all the time. But
so let's see here, absorbance. Oh, I see. Gotcha. So what we're going to do is let's take a look at calibrate. What Melissa is saying is she also, when working with software developers, developed this super cool, really neat mode called advanced mode, where you can look at two graphs simultaneously on top of each other, which is really what you want to do if you're looking at absorbance and fluorescence. You'd like to compare them both. So here we'll collect absorbance. There we go. So there's the absorbance spectra. Stop absorbance. And then go to fluorescence. And let's collect some fluorescence. I'll make this one, see, I'll make it 100 again. I'll make sure it's 405. I'm not going to calibrate it, and let's collect some fluorescence. There you go. Beautiful. Thank you for the suggestion, Melissa. That was great. So what you can see is there's the absorption spectra in blue and the emission spectra in red.
How did you get to the advanced mode?
So when you go to File, New Experiment, there's you can look at absorbance, transmittance, fluorescence, emissions, and then
down at the
advanced
bottom.
mode down at the
Got it.
bottom.
All right.
Yeah, and it allows you to toggle between different modes of collection. It's great. All right, so last thing that I wanted to show you. Let me hit stop here. Let's go to absorbance mode again. Absorbance versus wavelength. Just wanted to talk a little bit about plant pigments from algae. So here we go. So this right here is an isopropanol extraction from Spirulina, which is a cyanobacteria. And what's really cool is or I think is really neat, is if you take just one of these pellets here, they look bright green, this little cyanobacteria, and you toss that, you take one pellet, toss it in about, I would say 50 mils of isopropanol, take another pellet, toss it in 50 mils of water. What you wind up getting are two pigments. So this blue color, that's why cyanobacteria are called cyanobacteria, is because they have this brilliant, gorgeous blue color mixed with chlorophyll. What's really neat about cyanobacteria and other algae, this is actually a bacterium, so it's not really quote unquote eukaryotic algae, which is different. But anyway, we can still take a look at it. So let's just take a look at spinach again. Let's go back here, and where's my isopropanol? There we go. I'll calibrate the isopropanol. And then let's take a look at spinach, which is a terrestrial plant. Terrestrial plants have chlorophyll A and chlorophyll B. So chlorophyll A, these two peaks are chlorophyll A and chlorophyll B, along with that peak. Let's just take a look at the same isopropanol extraction using spirulina powder. What you can clearly see is lots more chlorophyll A. And what's interesting is this peak is not there, the chlorophyll B peak. It's all over here in this range. So this is almost all predominantly chlorophyll A. And what's really neat about this is if you take a look at Chlorella powder. So Chlorella is a green algae. Green algae are most closely related to terrestrial plants. They have chlorophyll A and chlorophyll B. Red algae, another eukaryote algae, only has chlorophyll A and then has like a red pigment. What's really neat also about this pigment, this is called, one of the pigments in here is phycocyanin, and it's a brilliant blue color. It's going to absorb mostly where you think it would look like a blue pigment, but it's also been important because it also fluoresces. The problem is I can't use this device to see its fluorescence. You would need to use the UV Vis fluorometer, but this compound will fluoresce, as will the chlorophyll in here too. So with that, why don't I stop and we'll take some questions, or we can look at some more things if you have other things you want me to take a look at.
Nicole,
go ahead.
I was just gonna say, Nicole just put a good question in chat. I don't know if you want to pull up your chat, John, or if you want me to read it to you.
I see it here.
Yeah.
Great question. There is a significant degradation, but what's really great is that it's mostly light related. So when we go to NSTA, I go to, normally when I would go to the National Science Teachers Association, I'm in the booth for Vernier, and I do a spinach extraction. It lasts all week long. And the reason why it lasts all week long is I extract it, I put it in a little glass vial like this, and then I store it in tinfoil in the darkened refrigerator, and it's good for a week. So you should be fine for a week using that. So probably not within days, it should last longer. The real thing is as long as students are comparing the same sample over like days. So if you made a sample on Monday and Monday's group is looking at that, as long as Tuesday's group is looking at the same sample, like you would see an aging, but they're still going to see the same thing, if that makes sense.
And also the covering it in tinfoil and in the refrigerator is very important.
Heat and light.
Note. Yes.
Heat and light. Just like we were talking about extra virgin olive oil, there's a reason why these bottles that you see over here, there's a reason why they're tinted and green colored and dark to protect it from light. And then they'll last a lot longer if they're kept at a cold temperature. Okay, the other thing I wanted to just show you really quick was we collaborate with 80 Instruments. 80 Instruments is a company that also does data acquisition, and they have a product called LT. LT is it's like an online curricular platform. It's really pretty cool. What's really neat about it is it's like the lesson plan exists on the web, but you also can do data collection with it. So our GoDirect sensors work with it. We have helped develop the biology collection. And if you take a look at the biology collection, there's a whole series of labs that are in there, but I want to preview one of the labs. So you can preview a lesson in LT, and the lesson that I want to preview is, of course, the visible spectra of plant pigments. And what's really nice about this lesson is you can use our GoDirect SpectroVis Plus with their online learning platform, but it's just got a lot more material in it, right? So it's got like all of this information about what chlorophyll is, other information about why it would be important. It's got little questions that you can have students ask, like identify what pigment gives the rusty red color of this blood orange. You could say anthocyanin, and then students check their answer. It's got an example spectrum for you to look at. You can formulate a prediction. It's a really nicely developed online learning platform that also supports data collection. And if you're interested, get in touch, let me know, and I can direct you to the folks at 80 Instruments. But this will also work with our spectrometer. It even has a little thing on how to calibrate using the spectrometer. And then they go through different food colorings and then looking at chlorophyll. So it's just another way to do this lab using an online learning platform called LT from 80 Instruments. All right. So any questions on that?
I do want to say are you guys, I guess, I'm sure, I don't know if there's any actual knowledge of, you guys are familiar with the Jove, Jove application, JOVE?
No, I'm not.
They use your stuff, and they use all your equipment in their videos. And I just wanted to suggest to everybody, we've been using it at the at PDCC for a while. It's a great learning platform. They actually have video textbooks, but most of their lab experiments, they do biology and all of that. They use your equipment.
Oh,
So,
great.
yeah, so a lot of the experiments that you have written, they literally are doing them from your book.
Great. That's terrific.
But it's a great site. The application is really, they've got quizzes too, but it's not quite as set up for lab like the one you just showed, but it's got some really good videos and things.
Yeah, we also work with Pivot Interactives, but spectrometers are not supported. And so that's another platform. There's just a lot of really amazing classroom management stuff that is in LT that we're never going to do, which is actually really cool. It's really like a course textbook with a bunch of online learning. Oh, well, it's a bunch of, it's kind of like having like a lab book that's available, can be available online, have sample data collected with it, but it's also got all the classroom management stuff in it as well.
Right. We'll have to look at it.
So other questions? How many of you have been teaching remotely or were teaching remotely over the last 2 years? Yes, no. There are some magic tricks that Melissa and I know about on how to do certain things. Data sharing is a really cool tool. Data sharing will allow you to share data from a computer to other devices. So there is a really cool thing that you can do. I don't know if I want to get into it, but if you're interested, please get in touch. You can email me at biology@vernier.com. Like I could take this data. Oh, you're all back in person fall and spring. What's really neat is I could take LoggerPro and I can send the spectroscopy data to graphical analysis, and then you could share it anywhere online. So it's just a really neat, interesting tool. And you also can insert videos. But if you're mostly working in person, I would say spectral analysis is probably the place to be. There's a lot that you can do with it. And just to remind everybody, spectral analysis works on every platform. So it works on Mac, it works on PC, it works on Chromebooks, it works on Android device and iOS device. That means even your phone. If you don't have a USB port, you would need to use the GoDirect SpectroVis Plus on Bluetooth, but it can do that. So with that, I think I'll leave it to questions. Are there any questions about this lab, other labs, things you'd like to see or interested in? Thank you.
So,
I see some people that I've met before. Like, hi,
I'm
Bobby.
sorry.
No, go ahead.
I have one thing I would like to suggest to you guys, and we've been trying to incorporate it. This is, I know I'm chemistry, but it's biology too, is things that are involving plastics. We've got, I actually did a lab over this in organic. We did where, like they have to check the densities, but that helps them split it up. But we're really trying to, and I'm sure everybody knows, and the kids are really up, they are, aware that this is an issue. And anything that you can come up with for some ideas of how to, I don't know, recycle things, make things, analyze things, and I think this would also have some applications for biology people too. But yeah, just a bug in your ear about that, because it would be really cool. I think it's really something we need right now.
You could do a lab with like bio magnification with the consumption of plastics that get stuck in, something like that. You could have, I don't know how you would do a lab with that.
I do know that we did have an activity where people were they're mostly microscopes and then a dissecting microscope to look at different types of microplastics, because microplastics are everywhere now in the environment. But yeah, an enzymatic degradation process where you can actually measure the amount of plastic. It's just, I think the plastics are so complex that might be a better thing to talk to a chemist about to see what type of assay you would want to use. But that could be really, really cool.
But
The
yeah,
other thing I wanted to remind everybody about is I have been using the spectrometer in wavelength mode, but I just wanted to remind the biologists out there that you still can use it to look at kinetics. So you can look at one wavelength, do your enzyme kinetic activity, things like that, or create your own standard curve as well and find out an unknown. So there are other ways that you can use it. It isn't just looking at spectra. It's actually a really complex, wonderful instrument. And I think that's all I've got, unless there's more chat or questions. They are going to post the recording from today. They're probably going to slightly edit it a little bit, and then they will, I think it'll actually be up on the web is usually what they do.
Yes, and everyone will also get a link to the recording when it's available. And in that same email, you'll get the link to the shared resources file and also a certificate of attendance.
Yeah, just to remember, you can always email support@vernier.com. There's chat at www.vernier.com. If you want to get in touch with a biologist, just email biology@vernier.com. Also, if you go to chat, you can find me probably there. If you see Melville floating around, you can say, "Hey, Melville," and that will ping me and I will come answer it. So yeah, those are all 3 great ways to get in touch with us. And thank you all for coming. This is really fun.